The eye and its connection to brain. Light enters the eye through an opening in the center of the iris called the pupil. It focused by the lens and cornea. The lens are adjustable and the cornea are not adjustable, and projected onto the retina, the rear surface of eye, which is lined with the visual receptor. Light from the left side of the world strikes the right half of the retina and vice versa. Light from above strikes the bottom half of retina and light from below strikes the top half. The inversion of image possesses no problem for the nervous system. The visual system does not duplicate the image, it codes it by various kinds of neural activity. Now I will explain the pathway of light through eye with the help of a mind map. Pathway of light through eye. The cornea bends or focuses the light ray onto the lens. Aqueous humor. It travels through the aqueous humor. Pupil allow the light into the eye. Iris controls the amount of light traveling through the pupil by expanding and contracting. Lens bend light ray to focus them onto retina. Ciliary muscle expand and contract to help bend and focus these light ray. Vitreous humor help to focus these light onto retina. Retina receive the light and process it. Fovea. Here the image is most clear focused. Photoreceptors, rods, cones. Rods. At night and low level light, these help us to see in black and white. Cones help to see color in bright light. Optic nerve transmit the image to brain. Visual cortex. Meaning is now added and the image is interpreted. Route within the retina. In the vertebrate retina, however, messages go from the receptors at back of the eye to the bipolar cells located closer to the center of the eye. The bipolar cells send a message to ganglion cells located still closer to the center of the eye. The ganglion cells' axons join together and travel back to the brain. Called amacrine cells from bipolar cell and send it to bipolar amacrine and ganglion cell. Amacrine cells refine the input to ganglion cell, enabling certain ones to respond mainly to particular shape, direction of movement, change in lighting, color, and other visual feature. One consequence of this anatomy is that the light passes through the ganglion, amacrine, and bipolar cell route to the receptor. However, these cells are transparent and light passes through them without distortion. A more important consequence is the blind spot. The ganglion cells' axons join from the optic nerve that exists through the back of eye. The point at which it leaves is a blind spot because it has no receptor. Fovea and periphery of retina. Fovea, pit, a tiny area specialized for acute detailed vision. Blood vessels and ganglion cell axon are almost absent near the fovea. Each receptor in the fovea connects to a single bipolar cell, which in turn connects to a single ganglion cell that has an axon to the brain. The ganglion cells in the fovea of human and other primates are called mid-jet ganglion cells. Because each is small and respond to just a single cone, each cone in the fovea has a direct route to the brain. Mid-jet ganglion cells provide 70% of input to the brain. Our vision is dominated by what we see in and near fovea. Receptors send a message to bipolar and horizontal cell, which in turn send message to amacrine and ganglion cell. The axons of ganglion cell from optic nerve which exist the eye and blind spot and continue to the brain. Visual receptors, rods and cones. Retina contains two types of receptor, rods and cones. Because of distribution of rods and cones, we have a good color vision in the fovea but not in the periphery. Both rods and cones contain photopigment, chemical that releases energy when struck by light. Rods abundant in the periphery of human retina, cones abundant in and near the fovea. Rods respond to faint light, low light condition. Cones respond to bright light. Rods see black and white and shades of grey. Cones essential for color vision. Color vision. Visible light consists of an electromagnetic radiation within the range from 400 nanometer to some 100 nanometer. Shortest visible wavelength as violet, longer wavelength as perceived as blue. We call this wavelength light only because the receptor in our eye are tuned to detecting them. Theories of color vision. The first theory is trichromatic theory by Young Helmholtz. The second theory is opponent process theory by Ewald Hering. The third theory is Retinex theory by Edwin Land. The trichromatic theory by Thomas Young and Hermann von Helmholtz. This theory was actually first started by Thomas Young and later on modified by Hermann von Helmholtz. So this theory is actually now known as the trichromatic theory of color vision or the Young-Helmholtz theory. According to this theory, we perceive color through the relative rates of response by three kinds of cones, each one maximally sensitive to a different set of wavelength. But receptors are not equally distributed across retina. He concluded that three kinds of receptors, we now call them cones, are sufficient to account for human color vision. In this image, we can see four different wavelengths of color. 3 for cone and 1 for rods. Response of short wavelength cone, response of medium wavelength cone, response of long wavelength cone. But this theory was incomplete as a theory of color vision because it was not able to explain the negative color after image. The opponent process theory by Ewald Hering. We perceive colors in terms of bird opposite, red versus green, yellow versus blue, black versus white. Bipolar cell that receive excitation from a short wavelength and inhibition from a long and medium wavelength cone. It can explain the negative color after image effect. The previous theory was not able to explain the color consistency, so the next theory came up, the Retinex theory by Edwin Land. When information from various parts of retina reaches the cortex compared information from various parts of retina to determine the brightness and color for each area. It can explain color consistency. Only works when entire view has been tinted. Color blindness and color vision deficiency. Complete color blindness, perception of only black and white. Color deficiency results because people with certain genes fail to develop one type of cone or develop an abnormal type of cone. In red-green color deficiency, the most common form of color deficiency, people have a trouble distinguishing red from green because long and medium wavelength cone have the same photopigment instead of different one. Thank you everyone for watching this video. Bye.